I'd like to introduce Eleanor Karlsson. Eleanor is a postdoctoral fellow at the Broad Institute, which is a big sequencing center in Boston. And Eleanor, I think given some of the previous questions about career and career paths and thinking about what you're interested in, I think Eleanor has a really interesting background. She attended Rice University, and you were a biochemistry and fine arts major. And it wasn't until toward the end of her college career that she decided to take a computer course, realized that she actually enjoyed computing and had some aptitude for computing. So I guess that led to a job doing data analysis at the Broad. So she started working at the Broad without an advanced degree, but then in the course of doing that work, realized it could be valuable to have an advanced degree and enrolled in a PhD program. Even though her PhD program was at Boston University, but her research was actually occurring at the Broad Institute. And just then, the Dog Genome Project was beginning. And she had a couple of interesting assignments to try and map some genes. And I don't know if you can ask her some questions during the discussion session. But again, she's been right in the middle of this sort of revolution in how these technologies have made identifying specific genes a lot easier. So with that, I'll let Eleanor take over.
Thank you very much for the wonderful discussion, and I'm really excited to be here today. I think this is actually a lot of fun activity. It's always fun to talk about dogs, and I was just actually going to start. So everybody asks me. I don't actually own any dogs myself, but my sister does own 2 dogs, Kaylee and Besco. So I put up their picture here, and I was just kind of wondering. So how many people in this room have a dog at home? Okay, see, lots of people. And then how many of you have dogs that are like in one particular breed? And how many of you have dogs that are like mutts or mixed breed dogs? See, so I think it's a really interesting question because it kind of shows how much dogs are really part of our life and part of our world. And it turns out that when we map genes in dogs, we actually do it very similarly to the same way we map genes in humans when we're looking for genes that that are changed, that you're born with, that put you at a higher risk of getting a disease. And so I just was going to go briefly over a little bit of the history of dogs. So dogs were domesticated from wolves, probably somewhere between 10 and 30,000 years ago. We don't know exactly when, we don't know exactly where, we don't know exactly why. It's actually a big field of research right now where people are arguing a lot about it. So I think over the next couple of years we'll start to find out more about that. But actually the breeds, even though people kind of always think about dog breeds as being, kind of the way dogs are, the breeds are actually, for the most part, not more than 100 or 200 years old. And that's when people took dogs and started saying, all right, we really want a massively huge dog that's really furry and got a shepherd kind of dog, or you wanted a dog that was really, really small and you got a Chihuahua. And while the breeds themselves are really cool, it kind of had the side effect where a lot of the breeds have very high rates of particular diseases because they often didn't use very many dogs to create the breed. And so if you only take 8 dogs to create your breed, 10 dogs, and one of them has a gene that causes cancer, when you have a whole lot of dogs in that breed, a lot of them are going to be carrying the gene that causes cancer. And this is why we started looking at genes, at mapping genes in dogs, is because we could use these breeds and look at these diseases that they had and see if we could find the genes that were underlying it. So this is a picture of our research collaborators on our work. We actually had these bandanas made a few years ago that said, I donated blood for disease research. And so we get all of our samples from pet dogs. And we tend to take a blood sample from them or a cheek swab. And you can see that the cute golden retriever has a little wrap around where they had the blood taken from them. Oh, actually, I wanted to find out. So have any of you had dogs that had different diseases or died from them or anything? Yeah?
My dog's kidney failed really quickly.
Anyone else? Yeah?
My dog has diabetes.
Another one? Yeah?
Me?
Yeah?
Same.
Diabetes? Yeah?
My Shelby had cancer.
Cancer. And so you kind of notice here that we've got diabetes, we've got cancer. It turns out that they get anxiety disorders and they get epilepsy and heart disease and kidney disease. And a lot of these things sound very familiar because they happen to be diseases that people get as well. And this is the reason why, back in 2005, we decided to sequence the dog genome. And when we talk about the dog genome, we actually sequenced the genome of a dog called Tasha. This is Tasha. She's a boxer. Just as a reminder, the genome is just the DNA that you inherit from your parents. You get half of it from your mother, half of it from your father. You carry a copy of your genome in every cell in your body, which is why we take a blood sample or a cheek swab. We can extract the DNA and the genome out of it. And so Tasha the boxer has a genome that's 2.4 billion bases long, so that's A's, C's, G's, and T's. So she's got those 2.4 billion bases in each one of her cells. And this is very similar to humans, 2.8 billion, mouse, 2.6 billion, so about the same size roughly. And if you compare two dogs from two different breeds, for the most part, and this is why the whole genome project works, we're actually getting the genome of one dog, Tasha, and we use that as a reference that we can compare all the other dogs against. And so this is because for the most part, your genome is going to be exactly the same at every single position. So for dogs, you see about one change every 1,000 bases between any two dogs, and with humans, it's almost very similar, about one change every 1,000 bases. And so for the most part, you've got the genome of the dog with that one dog. But of course, what we're really interested in is where is their genome different? And then once you've figured out where their genome is different, you want to know which of those differences are actually changing the way a dog looks, or possibly there's risk of getting a disease. And so the way we do this is we take a group of dogs that have one trait, so say here it's black dogs, and a group of dogs that has another trait, say white dogs, and we basically compare them. And what we want to know is there, any two dogs may have differences between them. And so you take any pair of dogs, you're going to see a lot of differences. But if you have a change that you only ever see in the white dogs and they're always different from the black dogs, that's an indication that it actually has something to do with the color of the dog. And then we do this using something called SNPs, which stands for single nucleotide polymorphism. Most of the genome in each of the dogs is going to be exactly the same, but every once in a while you find a position that varies between dogs. And so here you've got a SNP that's an A or a C, or a SNP that's a G or a T. So if you look at a whole lot of dogs from a whole lot of different populations, you're going to probably find about 10 million of these SNPs in total between all of the different dogs. And the way we map genes is we actually take a set of SNPs, and in dogs we tend to use about 200,000 across the whole genome, and what we're looking for are SNPs that correlate with your trait. And so in our example here, where we have a golden retriever who's not black but has pigmentation, and a white dog, the bull terrier, what you can see is we've got a SNP here where we've got it somewhat correlated. So the bull terriers always have a G there, but the golden retrievers are mixed between C's and G's. And then you have SNPs that are very strongly correlated, and here you have one that's completely different in the two groups. And what we think is that when you get those very strongly correlated SNPs, you expect that your gene that's controlling pigmentation or coat color is going to be sitting very close to it. So it gives you a very small region of the genome to go and search in. And this is what we've been doing, and we call it a genome-wide association study, and this is actually the results we get out of it. So all this plot is showing is basically along the bottom you have the whole genome. Each of the different colors is a different chromosome. In humans we have 23 pair of chromosomes, including X and Y. In dogs we have 39 pair in total. That's why there's so many different colors. And then we've plotted just the strength of correlation, and what you can probably see very clearly as you go across the genome is you hit that green chromosome, which is chromosome 20, and all of a sudden you get a really huge peak. A very, very strong correlation with coat color at that locus. And that's actually sitting right in a gene called MITF. And it turns out if you go and find out what people know about MITF, it's known to control pigmentation in mice as well. And actually in humans, it's linked to disorders of pigmentation, but also to disorders of hearing. So you often get deafness. And I don't know if any of you know this, but every once in a while, very rarely, but sometimes dogs that are white will also be deaf. And so this gene is actually a really good candidate for being responsible for that trait in dogs. And so what we've done as part of this workshop is actually looked at 3 different traits in dogs. And so I think some of the people in this room sent their dog's DNA in from a cheek swab. Can you raise your, yep, see, lots of people, cool. And so we got all of these DNA samples from all of these dogs from these cheek swabs, and we looked at 3 traits. We looked at short coat versus long coat, straight coat versus curly coat, and then we also had this trait called furnishings. I don't know why they call it that, but basically it's dogs that have like a hairy chin and a hairy mustache thing going on. And so the dog exercise out there, I think, had some information on all the different traits, but the one that we're actually going to look at here is the data from the coat length gene. So this is the short coat versus the long coat. And so just as you guys all got these handouts as you came in, and just as an example of what we're going to do with them is we're going to basically look for the SNP. You got 4 of them. There's A, B, C, and D, and we're going to look for the one that's most correlated with our trait of interest. And so what you're going to do is you're just going to go through and very simply count. And so in this particular little example that I'm giving here, you can see that the short-haired dogs have 4 Cs. So 1, 2, 3, 4. And then they've got 6 Ts, 1, 2, 3, 4, 5, 6. And then if you look at the long-haired dogs, you've got 1 C and you've got 9 Ts total. And you can see that there's a difference between them. So there's 3 more Cs in the short-haired dogs, and there's 3 more Ts in the long-haired dogs. So we'll just call it a difference of 3 there. And so you guys all have real data. I just want you to quickly count your letters in your data set. And then we'll put the data up here. Okay, so does anybody have position A? Okay, what did you get? For the short coat for the C and the T?
I got 12 and 11.
Let's see if this is, look at that. 12 and 8, yes. And then for the long coat?
I got 11 and 9.
11 and 9. So that's a very small difference. They're almost exactly the same, so it's just a difference of 1. Position B, anybody? Yes.
For the short coat, I got 15 and 5. And then long coat was 19 and 1.
Okay, yeah, so that's a difference of 4. It's a little bit more correlated, but it's still kind of mixed up. C, anybody? Yes.
15 and 5, 19 and 1, difference of 4.
Exactly. And then finally D, anybody? Yes.
17 and 3 and 2 and 18.
Cool. So that's a difference of 15. You will notice here that position D appears to be the most correlated by quite a lot. And so you've got a difference of 15 in the Gs and the Ts between the 2 groups. And if we actually go and look at the real data, you can see why we're getting such a strong correlation. And I should point out that what we actually do in our actual analysis of the data is we take those counts that we just generated, we turn them into a frequency, and then we calculate something called the chi-squared test, which some of you may have done in a statistics class, which gives us a p-value, which is what we actually put on those charts that I plotted out. But it's all based on those initial counts that we do. And what you can see here is that in the short-haired dogs on the left, most of them are GG, and occasionally you have a GT, but for the most part, all the dogs that are short-haired are going to be GG. And on the long-haired dog side, you can see for the most part, all of the dogs that are long-haired are TT, except for occasional GTs. And if we actually go back to our cute little dogs that people sent in, these are the short-haired dogs. Anybody have a hypothesis as to what their genotype is going to be? Thank you. Yes, GG. The long-haired dogs, exactly. I'm sure that there was a dog that wasn't a golden retriever in there that was TT, but whoever you were, you didn't send in a photograph, so. But the really interesting thing that this also gets at is just the complexity of genetics, and this has actually come up in the cancer discussion and in the autism discussion, which is that all of these dogs, which are all short-haired, these guys are all GT. But actually, we also have this dog, who's very clearly long-haired, and is also a GT. And so even though we've got a very strong correlation, and this SNP is actually sitting in a gene called FGF5, which when they've looked at it in mice is connected with hair length, it's clearly not the whole story. And you might hypothesize that this dog has another gene in the genome or a different change in FGF5 or a completely different gene in the genome that's influencing the length of its hair. And so at this particular position, it's not having a big effect. You know, it has long hair even though it looks like all of the short-haired dogs. And so, yes, a scientific mystery. And I'll point out a very cute dog, whoever's dog it is. And so this is kind of what we've been doing as far as actually mapping traits within the dogs that we've been looking at. But of course, the other thing that we looked at with the data set that you guys, the samples that you sent in, was we actually went back in time. And rather than worrying about what was going on with these dogs down here, kind of the breeds and things, we actually went back and said, all right, what actually separates the dogs from the wolves? So wolves are not dogs. You would not want a wolf living in your house. They tend to be very violent, and they tend to eat people and small children. And so clearly dogs and wolves are different animals. And you know, there's a lot of hypotheses as to why dogs started living with us. They might have been eating our garbage. It's possible they helped us with hunting, or they warned us when other people were coming around. But the question is, can we actually compare dogs to wolves and find out where those differences are? And so what we did is we took, so this is actually it's plotting a different measurement, but it's the same idea where you have all of the chromosomes across the bottom, and what it's measuring is just the dog-wolf difference. And so we're actually able to do this now because of all this genome sequencing that we've been talking about over the past couple of days. So they could actually get genome sequence from a whole lot of wolves and from a whole lot of dogs, compare their genomes and say, where are they the most different? And so what you can first of all see, as compared to that coat color picture that I showed you earlier in the presentation, is there's a lot of different peaks in here. So there's a lot of different places in the genome where dogs and wolves are different, which you would expect given how different they are. And the one that we're actually going to focus on today is this one right here. So this black peak here, right here, is at a gene called the amylase gene. And the amylase gene is involved in digesting starch. And what they found is that when they looked at the wolves that they'd sequenced and said, okay, what's going on at the amylase gene, the wolves had exactly what you would expect. As I said, you get one copy of your genome from your mother and one copy from your father, so you have 2 copies of each gene in your genome normally. And when they tested 35 different wolves, and every single one of them had 2 copies of the amylase gene, exactly what you'd expect. But when they tested 136 dogs, what they found out was that every single one of them had more than 2 copies and up to 30 different copies of the amylase gene. And when you think about it, why on earth would you duplicate a gene? Well, the more copies of the amylase gene you have, the more, the greater the amount of the amylase protein you can make. And the more protein that you're making, the more starch you can break down. And so basically, the fewer copies you have, you're going to digest starch worse or less efficiently. You may not be able to get as much energy out of the food when you're eating starch. The more copies of the amylase gene you have, the more, the better you're going to be able to digest starch. And probably what happened is that when dogs started living with us, the first thing they needed to do in order to exploit us as a resource is learn how to eat our diets. And so they actually evolved the ability to digest starch much better than wolves can. And interestingly enough, there's other researchers that have looked at the amylase gene in humans and found out that humans compared to chimpanzees have extra copies of the amylase gene, probably for exactly the same reason. And so dogs and people actually get many of the same diseases. And I think that the nice thing about the amylase story and the thing at the beginning where I asked you guys about who owns dogs is that not only are they getting a lot of the same diseases, cancer, heart disease, diabetes, anxiety disorders, epilepsy, kidney diseases, they're also sharing our environment. They're drinking our water, they're eating our food. When they get sick, they go to the vet, they get treatment. When they get cancer, they get chemotherapy, they get radiation. And so they're actually living very similar lives to people. And so when you start thinking about the interaction between genetics and environment, that starts to get very interesting. This kind of gets to the point that dogs share many of the same diseases as people, as I said. And what I've put down here is actually a plot just showing, this is actually the most recent data from our bone cancer study. So dogs get a disease called osteosarcoma or bone cancer. Greyhounds get it a lot. About 1 out of every 4 greyhounds will die from bone cancer. And bone cancer is also very common in humans, and it looks almost exactly the same in humans as it does in dogs. In humans, it often strikes teenagers during the period of time when they grow very quickly. And often in dogs, it often strikes large breed dogs. And what we saw when we went across the genome is, once again, we have several different peaks, but we have a very strong one there on the green chromosome. And that's a gene called CDKN2A. And if you go and look in the hallway on the big cancer poster that's out there on chromosome 9 in humans, you'll find CDKN2A listed there as a cancer gene in humans as well. And so we map genes for diseases like cancer in dogs just the same way we map genes for coat color, for hair length, for curliness, for furnishings. And it's actually a really exciting way to kind of look at what's going on in people as well. So any questions out of all of that? Yes?
I know you were giving your introduction, it talked about your experience with coding and how computers helped you with some of your research. Can you talk a little more about that?
I think that's a really good question, and I think it came up during the discussion. So I took one programming class as an undergrad. My last year, I had a bunch of friends who were CS majors, and I was really annoyed because I kept on talking about computers and I couldn't understand them. So I took a programming class, and it turned out that I really enjoyed it. And at the same time, I had just started working in research labs over the summer. And I was kind of working in a lab that was studying the cell cycle, and they were kind of studying one thing at a time, and they figure out that this thing went wrong in this cell at this time point. It was all very specific, and I couldn't figure out how you were answering the bigger questions. And at the same time, the Human Genome Project was just getting started, and it was kind of apparent that nobody was going to be able to sit down with a piece of paper and study 2.8 billion bases in the genome. And so at that point, it was kind of all came together at the right time, and I got a job as a programmer on the Human Genome Project. One of the interesting things about scientists is that if you're smart and interested, they'll often hire you to do jobs that you don't know how to do yet. So I'd taken one programming class, and it was in one computer language that I've never used since then, but they took me on anyway, and I had a lot of fun working on the human genome. That was down at Baylor College of Medicine. And so I actually took a few years off then because I didn't know exactly where I wanted to go, what I wanted to do next, and worked as a computer programmer, and that was when I found out that I actually didn't like computer programming without the science. For me, it really is a tool to analyze the genome, and got back into it again. But it's really quite amazing right now because since then, our ability to generate data has just increased so fast. I mean, you can sequence the DNA now of every single, you can start sequencing many different cells out of a tumor and start seeing how the tumor changes over time. You can start sequencing the RNA and figure out which genes are turned on and off at different time points. And every time you do that, you're creating a huge amount of data, which is an incredible resource that we have absolutely no idea what to do with it yet. And that's going to actually be the really big challenge over the next 10 or 20 years, is figuring out how do we take, now we have the ability to generate this vast amount of information, but how on earth are we going to actually use it to figure out things that are relevant to human health? Yes.
Cool. It's been seen in boxers that they tend to get cancer very early in their lives, and most don't even live past 10 years old. Is that because of something in their genome, or is that just chance?
So the short answer would be yes, and not boxers in particular. But the interesting thing with dogs is it gets to that question that kind of came up earlier, which is environment versus genetics. And the question with autism, where they look at twin studies to figure out how genetic a disease is based on how often an identical twin inherits it. And with dogs, we're kind of doing the same thing, except that we're looking at the breeds. And so with the bone cancer study I was talking about, 1 out of every 4 greyhounds is getting bone cancer. That's not true in all the other breeds. And so that's a very strong indication it's got something to do with the genetics of the greyhound and not with environmental factors, because otherwise you would expect to be seeing it cropping up in all the other dogs as well. And so a lot of dog breeds do have problems with particular diseases, and because of that, their lifespan gets affected by it, because they're dying earlier. But I don't think that, I know the boxers have also had a big problem with cancer, but there's a lot of other breeds that have issues with cancer as well. So I wouldn't say that it was specific to them, but it is something genetic that they're born with. Yes.
Have you made any interesting programs to analyze the data that you have in your project right now?
I've made many programs, whether they're interesting or not, it's a matter of opinion. So I really enjoy what I'm doing. I mean, I love doing science. I think it's one of the most exciting things anybody could be doing, because you're trying to answer questions that nobody else knows the answer to. But because you're trying to answer questions that nobody else knows the answer to, you're having to create all of the tools that you use as you go along. And so there's 2 ways that I approach it. I write a lot of my own computer programs to analyze the data. I spend a lot of time thinking about the data, trying to figure out what kind of patterns it is that I'm looking for in there and how to detect them. But I also, and this is the other really fun thing about science, is it's a massive collaboration between lots and lots of different scientists. And so I spend a lot of time, everybody thinks it's very funny, but when I run into a really difficult problem at work, I often go to the coffee machine and get coffee and hang out there for half an hour, because inevitably somebody will wander by and I can ask them and they'll say, oh yeah, I had exactly that problem. And this is the way I dealt with it. Or I heard this talk last week where they were doing this, and I can then go find the next person and talk to them about the problem and kind of bring everything together.
What language have you worked with?
As far as whatever is most useful. I mean, I've done, I can't even remember what the first one was, but a lot of Perl, R, C, Java. Somebody eventually is going to convince me to code in Python because it's the cool new thing, but I haven't had to do it yet. Yes.
What else are dogs' DNA going to be used for besides this one activity? Is it going to be used in future programs you're going to do? Or?
So what was done for this particular project? So as far as this project goes, we looked at 3 different parts of the genome that were related to these 3 traits that I talked about, and then also the amylase gene. And so it was kind of really designed for doing this project of how do you, just an example of how we map things in dogs. And I was actually really excited about the amylase poster that's out there where they looked at, all the different dogs that you guys sent in and where they fell on the spectrum, because you could see that it didn't have anything to do with what breed the dogs were. It was kind of spread out all over the distribution, which is kind of what you would expect to see, since we think it goes all the way back to when they were first domesticated from wolves. And so it's actually really interesting from that point of view, but it's also just a great example data set. When we look at dogs for the genetic studies we're doing, we try to generate the data across the whole genome. And this is where the computer stuff gets so important, is because the really wonderful thing about generating a whole genome's worth of data is that not only do you have the thing in there that you're interested in, hopefully, but you also have the rest of the genome, so you know what it looks like in the places where there isn't anything interesting. So you have both the correlated piece and the pieces that are completely uncorrelated, and so it gives you a chance to compare. If you only have the thing that you're interested in, it's hard to know how interesting it is unless you have everything else to compare it to. Yes?
Are there other programs going on for other animals?
Yes. So dogs were one of the ones that we started with first, just because, well, they're the coolest, of course, and but also because they're the first animal to be domesticated, and there's also this incredible, what we call breed structure, which is that there's these 420 different dog breeds, which were very important early on when we couldn't do genome sequencing as easily as we can now. Now that we can do genome sequencing, we can start looking at a lot more different kinds of dogs. But there's also a big cat genome project. There's Stickleback is this really cool fish that they recently published a paper on. There's lots of different animals that they're looking at. And it's going to be really interesting to see, different animals are good models for human health in different ways. Like I think, the guinea pig is really good for immune-related stuff. And so it really kind of depends on what questions you're asking, once again. Yes?
Do dogs of a mixed breed tend to be more or less prone to dogs of a single breed?
That's a really interesting question. And I actually thought about this recently, and I think that for dogs that are in a single breed, they tend to be prone to very particular diseases. If you look at a group of dogs from any, just a crossbreed of dogs, they're not going to be getting one disease overwhelmingly, but they may get more different kinds of diseases. And so it'll be a more diverse spectrum of diseases, but they still have to, die from something eventually. So that's a very depressing way to think about it, but there you have it. Yes?
I'm kind of curious if there's a possibility of breeding dogs with wolves and seeing if they, what kind of gene can cause them to become more domestic or like,
Yeah, no, that's actually, it's an interesting idea. They actually did something like this in the other direction with foxes in this very famous study that they did in Russia that I think has moved to the US now, where they took foxes and domesticated them the same way that we presumably once did with wolves, just by figuring out which ones were easier to handle and would deal with people. And it took them decades, but not more than that. So it wasn't, my theory is always that it couldn't have taken that long to domesticate dogs from wolves, because wolves are really dangerous and we wouldn't have wanted them hanging around that much. But that said, that's an open question. We don't actually understand the process that much. I'm not sure whether breeding dogs back to wolves would answer that question, although it might, because you could figure out which dogs were more wild than which other dogs and which genes were connected to it. So it's actually an interesting idea. Practically, there's probably problems with it. I don't know what you would do with the half dog, half wolves once you made them. And a lot of times with genetics studies, you have to make like hundreds. So there'd be a lot of dog-wolf hybrids floating around to find homes for. So I think I'm supposed to end now. Sorry. I was just about to take another one.